Hi everyone, this is Wasim. We are now on tier 2 answers, and so we will get started. Case 1 is a massive hemorrhage protocol is activated on a woman being brought in by ambulance. As soon as she arrives, the tube of blood is drawn before any red blood cell transfusions take place. You begin with the RHD typing and find weak plus reactivity with an RHD reagent. The patient is in your blood bank record from a few years earlier and is typed as RH negative. So if you've done the tier 1 question and answer session already, these first two questions should be fairly easy for you. What RH type will you issue to the patient during the massive hemorrhage protocol, RH positive or RH negative? Remembering that for the RH type, the magic number in the MHP toolkit is 45 years old for women. So what we have is a woman of an unknown age, and so because we don't know the age, we would issue RH negative until we know her age to be on the safe side. In terms of actually knowing the age, so if we assume that her age is 65, what would we issue? Again, with her age being now greater than 45, we would issue RH positive. So really the main difference between these first two questions is unknown age. In that case, you're always going to be the most conservative and protect the patient, especially in this case for women. So you give RH neg. For a known age that's over 45, you can then follow the process and then issue RH positive. So now you start thinking about this in the lab, and you incubate the patient's red blood cells with an RHD typing reagent longer, essentially doing a weak D test. So you take your RHD reagent, you add it back to the red blood cells, and all you would really do is you just incubate it longer, and then you read it at essentially the IAT phase after your 37-degree incubation. Some of the reagents are blended and some of them are not. So if you have a blended reagent, you'd read it at 37. If you don't have a blended reagent and it's simply just a monoclonal, you'd read it back at immediate spin again. So the blended reagents have an IgG and IgM component, and then the monoclonals usually are just IgM only immediate spin read. But these reagents are nice because you can read them at immediate spin and at IAT. So depending on what reagent you'd have, you then would reread it and you'd look to see if there's agglutination in the tube. So you now do that and you see that the reactivity is unchanged at week plus. You now try 3 other typing reagents, all week plus as well. And now we ask, what is the serological classification or explanation just based on that alone? So let's look at our answers. Weak and partial D, that one's already kind of out. You'll see why with B and C. Previously transfused before the phlebotomy was drawn. Now theoretically, if the patient was transfused before, they could potentially have a weakened D. So if they are Rh positive and they get transfused a massive amount of Rh negative blood, right? If we didn't know her blood type or she was less than 45, she may have gotten a lot of Rh neg and she may look weakly positive. However, if you were to incubate this for a longer period of time, you should normally see it go from weak to 1, 2 plus. It shouldn't just stay so weak that you can't see anything. So unlikely to be from the prior transfusion. Now we have two options, weak and partial D. And in the next slide, I'll talk about how you should think about them serologically and kind of molecularly in terms of the way they express their protein. But for just the sake of the answer, the answer really is going to be weak D. And I'll show you why in a moment. Now, for those of you that know about weak D, you're probably yelling at your screen right now because most weak Ds, when you incubate them longer, they go from like weak to 1, 2 plus, sometimes 3 or 4 plus if you're really, really lucky, which is very uncommon. But in this case, I made sure it didn't change. So there's a reason for that, and we're going to talk about that on the next slide. So I'm going to tell you a little story about the RHD pizza pie. This is the way it was described to me when I was a fellow, and I've never forgotten it. I think it's a really good way to actually talk about it. So if we think about RHD, as a pizza, and this particular person has just a normal RHD, so they're wild type, was what we'd call them. And we think about someone who's got an RHD, some sort of weak D variant, and then an RHD that's a partial. The difference is that with a weak D variant, let's say this one is a large or XL pizza. The RHD of the weak variant would be maybe a small or medium pizza. The only difference between this pizza and this pizza is that the RHD just has less. So if the red blood cell is decorated with RHD, and it's excessive. The normal red cell wouldn't have that much, but let's say it has a ton of it on there. Oh, geez. Let me erase this. The RHD for a weak D person would have less, which is why it's weaker. And that's why I said if you know enough about a weak D, you're probably yelling at me right now because I made that question a little more tricky than it needed to be, because if you incubate a weak RHD, it usually goes from weak to 1 to 2 plus, which is how you know it's a weak D. But in this case, I didn't, because I tried to make it closer to reality. The reason is if the patient was transfused Rh negative units, right? Excuse me, let me do this again. If the patient was transfused Rh negative units and they were Rh positive, they were already going to have a weakened agglutination from that, and then coupled with a weak D, you potentially might not see it if it's weak, if it's too weak. So that was the little curveball that I threw for the tier 2 question. Now for the partial D, it's a little bit different. Rather than size, it's really about missing a piece of the pie. So it's as if someone walked by, grabbed a slice or two, and walked away, and now it's it's pizza. If I were to ask you what it is, you'd be like, it's, even though it's an ugly pizza, you'd say it's a pizza. And if I were to ask you, are they different? You would say, yeah, I can tell there's a piece missing from it. And that's actually how the body also looks at it. The body doesn't see it as a true RHD. It sees it as a new protein. And as a result, that's why these individuals can make anti-D, because their pizza is not the same as this pizza, and the body sees it as different. And RHD and the partial Ds are all a little different. Sometimes you've got multiple pieces missing, sometimes one piece, sometimes you've got only half a pizza, and the variants will all change the protein expression. But as a result, for the most part, the partial RHDs are viewed as different from the wild type RHD, which means that they make anti-D. And so there are lots of partial Ds. And the other thing you should know about it is some of the variants that are out there not only change it from a extra large or a large pizza, sometimes it's a medium pizza with a slice taken out of it. So they come in all sorts of sizes and shapes. Now, what's unique about this is if you were to take an RHD typing reagent, okay, and remember I told you there are two types. There are the only monoclonals, and then there are the blended. So if you took the monoclonal, like the single monoclonal that's IgM only, and you looked at the IgM IgG version, when you incubate the IgM, you might get 0 reactivity, you might get 3+ reactivity. It all depends on where the antibody binds. If the antibody were to bind right here, right, it looks like a normal RHD. If the antibody tries to bind in this missing pizza spot, it has nowhere to bind, and so you get no agglutination. But here, you might get 3 or 4+. So it all depends on the antibody you use. So if you move around and use different just immediate spin reagents, you'll see reactivity all over the place with a partial D. Now, one of the beautiful things about the blended reagents is the IgM and the IgG actually look at different parts of the protein. They're made to be different antibodies. So if let's say our IgM looks right here, and our IgG were to look in the same place as this other antibody, we would have potentially, let's say 3+ reactivity here and no reactivity there. Usually the opposite happens, and your IgM is negative, and then you go to incubate it longer, and all of a sudden you get a 4+ reactivity out of nowhere. And so the weakened partial Ds are different in that if you incubate longer with the same antibody, the weak D will get stronger, but the partial D won't, because it's either binding or not binding. With a partial D, when you swap up the antibody, you should get binding. If as long as there's enough of the pizza pie there for it to recognize, it should be fine. And so remember that partial Ds, pretty much all of them will form have the potential to form an anti-D, but for the weak Ds, they don't all form anti-D. It's essentially weak D1, 2, or 3 do not form it, and then 4.0 and above are thought to form anti-D. And really you want to be especially careful with the 4.0 and the 4.1 with pregnant women or chronically transfused individuals. You want to get them Rh negative blood because they're going to be exposed to it more often than your, let's say, average patient who might show up and just get a transfusion once in their life. Because in a pinch, you could argue that because the antibody forming potential of a weak D 4.0 or 4.1 is still, it's not unknown, but it's not as risky as some of the other weak and partial Ds. You could argue that for someone, when your inventory is short, you can give them Rh positive blood and they're not likely to form an ATD and it's safe. But in a chronically transfused individual who is like a sickle cell or something, and they're weak D 4.0 or 4.1, you definitely want to give them Rh negative blood because you don't want to risk it. Or, for example, also a pregnant person. A woman who is carrying child, you don't want her to form ATD and she is weak D 4.0 or 4.1. You'd want to give them Rh negative blood. So the newest guidelines state if you're chronically transfused or if you're pregnant, you and your D looks off, for example, let's say it's 1+ or 2+, you should get the RHD sequencing done or some sort of RHD variant typing done in order to identify whether or not you've got a weak D status that potentially could cause you to form ATD or a partial D that could also cause you to form an ATD. So remember, pregnancy and chronically transfused individuals are the ones that we really want to chase down these weakened partial Ds on because they have the chance of forming anti-D, and it makes our life more difficult to find blood, and it makes their lives more difficult, and also potentially harm them with regard to the fetus if they are Rh positive. So we know that something is off, right? So we send it for our in-house CBS RHD sequencing, and it comes back with RHD01, which is a wild type, means normal pizza pie, nothing else, nothing wrong. And that obviously doesn't jive because we know serologically it's not making sense because it's a weak reactivity that's not changing with increased incubation. And so we send it off for RHD sequencing by next generation sequencing at an outside lab that has longer read lengths and can do the pretty much daisy chain the whole gene because it's fairly long. So based on their genotype, what would you expect of the expression of RHD on the red blood cells? And I gave you this hint because I know many of you are not as comfortable with genotyping since it's not as common in Canada with regard to next-gen sequencing. But don't let the genotype scare you, right? Let the serology guide what it is. And with RHD and CE, and really any next-gen sequencing, you always want to fold in the serological findings back to make sure that it all makes sense. So I'm not going to even look at the genotyping for a second, and we're going to answer it just based on serology. We know that if we think this is a weak D, it won't have enhanced. There's no way it has enhanced D, right? That doesn't make sense. It's not a partial D because we tried different monoclonals and the reactivity didn't change. Therefore, it's not missing a slice of the pie. The pizza pie was just smaller. And we know there's not an absence of D because we're seeing weakened expression. So answer is C. And when we now go back and look at the genotyping, what we have is this guy right here. So what it's saying is that we've got two alleles, okay? So DNA strand 1 from mom, DNA strand number 2 from dad. Mom has no D gene, right? It's been deleted. Let's just assume mom gave her no D gene, which means one of her genes does not produce a D. Now, let's say dad, dear old dad, gave her a duplicated exon 3. That's written out here. So in her gene, she's got exon 3, and now it's been duplicated. She has another one of them. And what this does is, as the protein's being produced from this, right? So we've got our little machine here. It's going to run along the DNA and produce the protein based off of it. As it's reading forward, it's going to see the double blue, light blue squares, and it's going to repeat itself, right? But that means the protein's not going to be the normal protein, right? It's going to be slightly longer. It's going to fold slightly more oddly, and it's not going to quite fold the way it should and be presented the way it should. It also means that it's possible, based on the way the duplication happens, you could have an early stop codon. You could form a protein that doesn't fold right and never makes it out to the surface, or makes it out to the surface of the red blood cell less. So you just have less of it out there floating around. So this quote weak D actually has two bits to it. It has an altered protein, so the protein's not the same, right? It's got an extra chunk to it, and there's also less of it on the surface. So it's kind of a mix between, for the lack of a better way of describing it, it's like someone shoved an extra slice of pizza into your pizza pie, right? And so you have overlapping pieces and it doesn't quite look right, but you can kind of eat it and it's all good, but you know that it's not also the same either. So it's a hybrid of like a partial and a weak D in a sense, but because the protein is altered, the red blood cell has the machinery of the red blood cell has a difficult time getting it out to the surface and presenting it. That's at least the thought behind the mechanism of this exon 3 duplication. So this is a paper looking at a similar exon duplication within exon 3, and I'm going to go over a little bit of their data because I think it's interesting. So they did a Western blot looking at the protein expression, and they expressed RHD exons 2 through 4 and 6 through 8. And what you can see is this is an RH positive individual, RH negative individual, and this is our person with the exon duplication. This is their control, so you can just ignore that. And when you look at a normal person, they've got one band here of the product when they have exon 2 through 4 intact, and then 6 through 8, they have this protein. In an Rh negative person, as you'd expect, there's nothing there. And when we look at our special little individual that has the exon 3 duplication, their protein size is different and it's much larger, right? And this is still faintly there down here, but you also get all of these weird products with a big one right there. Exon 6 through 8 isn't touched, so that protein product is fine, but you can imagine that if your regular RHD protein, looks like this, the folding of this one is going to be different and a little bit bigger because of the extra duplication. So they're not going to look exactly the same, and therefore the potency of forming an anti-D is actually quite possible with this exon duplication. And remember, I told you, because this thing is a little bit larger and not folded quite right, when it's sitting inside of the red blood cell, and you got this protein that's a little bit bigger, there are shuttling proteins that take them out to the surface of the cell, but those shuttling proteins look for very specific regions to kind of latch onto and yank them out. Because it looks a little different, it actually doesn't make it out as easily, and therefore not as many make it out to the surface. And so you've got fewer of them. Oops, wrong color. You have fewer of them out on the surface compared to a regular RHD, and they're a different shape. So that's why I said it's kind of a hybrid of a weak and a partial D in a way. And as the manuscript states, it showed that transcription is severely altered. The amount of transcript varies from individual to individual. So a person with an exon 3 duplication, one person may have a lot more protein on the surface than another. They don't really explain why that is. The antigen density is a huge range. You can see about a 1,200 to 6,000 range. 1200 would be fairly weak when you're talking about agglutination, and they all showed weak positive agglutination at the immediate spin phase. So even with 6,000 copies on your surface, you still only get a weak plus agglutination. And we've kind of already answered this. Would you expect someone to form an anti-D with this exon 3 duplication if they were exposed to Rh positive blood? And the answer is yes, because again, they're kind of a weak and a partial D. And remember, some weak Ds, especially anything above a 4.0, can form anti-D, and then essentially almost all partial Ds will form an anti-D. And because it kind of has this hybrid and it's a new protein, it can easily form an anti-D should they be exposed to Rh positive blood. Okay, our second case is a 25-year-old man who presents to the emergency room with profuse gastrointestinal bleeding. He has 3-year history of colorectal cancer and is receiving chemotherapy in a week. In order for 2 units of red blood cells is placed, and you also receive a sample for a type and screen. So let's take a look at our forward and reverse typing for our ABO for the type and screen. By forward typing, they look like they're AB, and the text noted an asterisk on the anti-B forward typing reagent, and they said there's 2 cell populations. On the reverse typing, the individual looks like group A, since they have anti-B in their plasma or serum. And by the RHD typing reagents, we've got 2 of them, 3 plus and 4 plus, so they look like they're Rh positive. The DAT looks like it's negative by polyspecific, therefore negative overall. And so really what we're dealing with is a discrepancy in the forward and reverse typing. Forward typing looks like in a B individual, and reverse typing looks like an A individual. So in this particular case, we need to figure out, and again, we have to pick a direction. If you did the tier 1 questions, you'll know that you need to pick a direction about which one is the incorrect, quote, incorrect one, and then travel down that pathway. So it's either forward or reverse typing being incorrect, and then we have to decide extra or missing antigen or extra or missing antibody and travel down that pathway. All right, so where is the serological discrepancy? So we kind of discussed this on the last one. We have a forward that is AB positive with 2 cell populations for the B reagent. And for the reverse, we have an anti-B, which means that the person is group A. And so you remember I said that based on the tier, if you did the tier 1 question and the answers, you know that we have to pick a direction. And from my perspective, this one's okay. And the thing that kind of jumps out at you, from me at least, is the fact that there's 2 cell populations with the B. And that seems to be discrepant. So I would say that the forward typing is what's off. So we know that the RHD and the DAT, the controls and everything looked good. So there's no reason to ever question the discrepancy there. And so then you're stuck between forward and reverse, and it's the forward because there's a discrepancy in the B cells being typed. Based on the patient history and serology, would you expect this discrepancy to be clinically significant? So this is a tough one because you have to really think about whether or not this discrepancy would cause any issues and whether or not it's going to cause harm to the patient. Now, the history of the patient tells you that they have colorectal cancer, and that's the key to all of this. So people with colorectal cancer can have something called an acquired B. And we'll discuss more about this in the coming slides. But in essence, this phenomenon of having this acquired B when someone's a group A is actually not clinically significant. And so you'd have to key into the fact that they have colorectal cancer and that there are 2 cell populations on the B reagent and that they're likely group A, and that the discrepancy is due to a tube-based phenomenon. So I'm going to apologize if my audio sounds a little weird. My headset gave out, so I had to switch over to just the computer audio, and I'm in tablet mode, so it may be a little off. So we've got the next question, number 3. You decided to run an additional monoclonal anti-B. Based on these results, what is the cause of the discrepancy? So remember, these are new anti-Bs, so we've got Immucor, Biorad, DBL, and Ortho. And we run it against the cells again. 3 plus, no agglutination, 2 plus, 2 plus. And this should remind you a little bit of the RHD story of that pizza pie that I told you about. And so if you think about it, if we were to have a B subtype, or excuse me, a B subtype, most of those act like more similarly to a weak D. So you tend to have weaker reactivity with all of the monoclonals you would use. An acquired B actually can look more like a partial D, because you'll have 4+ reactivity or 3+ reactivity, it'll go away and come back again, and it's because of the fact that the monoclonal antibodies look for a very specific epitope, and the acquired B epitope, which I'll show you on the next slide, is either there or not there, so it's either detecting it or not detecting it. Weakened B, if we had a weakened B, it'd be more similar again to, for example, a weak D, where it'd be a weaker reactivity, you'd incubate it, and it would get stronger. So we can get rid of this one and this one. And then hematopoietic cancer. So hematopoietic cancer can cause DNA disruptions through various mechanisms. For example, with MDS, you get hypermethylation, which shuts down genes, or you can get DNA degradation from the fact that they're replicating and DNA gets not destroyed, but it gets kind of messed up during the replication process. And so when that happens, usually the transcript, so for example, the B antigen, or the enzyme that would produce the B antigen, is malfunctioning, and therefore everything should be off, not just one monoclonal. So it shouldn't be this, because you'd either have weakened or no agglutination with this across the board. So we're left with one thing, an acquired B. So let's go on to the actual science behind it, and let's learn a little more. So how is it that a group O individual can now look group B? And this really comes down to the structure of the A and B antigens. So when we're looking at the A and B antigens, we first have to know the structure. So the fucose that's here denotes group O, or the H antigen, and the sequential addition of a sugar on the galactose here is what makes it group A or B. So remember, sugars are added sequentially. So if you were, for example, a group O individual, you wouldn't have any of this. You'd only have the fucose attached to the galactose. If you are a group A person, you'd have the addition of the fucose first and then the GalNAc second. If you're a group B person, fucose goes on first, galactose goes on second. So if you look at it structurally, these two sugars are actually very similar to one another. This is a N-acetylgalactosamine, and this is just a galactose. So they're similar enough such that when certain bacteria are infecting humans, they have an enzyme called deacetylase. That deacetylase can remove the N-acetyl group, or I should say the acetyl group, leaving behind an NH3, which then can look similarly by monoclonal antibodies. This is purely an in vitro phenomenon. It is not like, even though the human body can form antibodies that are, quote, group B as a result of this, the fact that we see it in tube is more of a, due to the fact that we're using monoclonals that are very specific for a certain epitope. So let's take a closer look. So we have the group A individual here, and let me highlight. This is the N-acetyl group, right? And remember, for a group B person, this part that we see here doesn't exist, right? This is the equivalent of it here. So they look similar except for this N-acetyl group. And what happens is the deacetylase here comes in and cleaves here. It essentially attacks and causes the oxygen group to shift over. So we're left with this amine group. And now when we have monoclonal antibodies coming through, They can't tell the difference between these two and they'll react. Now remember, in the last question, we saw monoclonals that reacted and didn't react, and it's because of the epitope that it's searching for. So some monoclonals will recognize um, this hydroxy group is the same as this amine group, and some of them will not. And so if you happen to use a monoclonal in your lab that were to do this, you would see it drop out and come back potentially if you had another monoclonal to use. You'll see on the next few slides as I talk a little bit more about the history of this, it's very uncommon to see this, which is why I included it in this question, because when Neural saw this, it's very uncommon because the monoclonals that used to be the main culprits of this are no longer used anymore. So when you see it, it's actually something you should really pay attention to because it's not something you'll see very often. I've seen maybe, I think, 2 of these in 8 years, and I was at a huge national reference lab. Now, I told you before that the deacetylase can be released by specific bacteria, and this particular question is asking you which bacteria does it. And the only answer is E. coli. There are a couple others that can do it, like Clostridium species can release deacetylase, but the most common one is E. coli. And often we see it in people with some sort of GI cancer. The reason is E. coli is one of the healthy bacteria sitting in our gut. And so this is our gut. If you end up having cancer, for example, and it eats away at the lining, well, the blood vessels that are running along it can pick up the bacteria. And so those bacteria then get through the bloodstream and move on, and they can release their deacetylase, which then is, of course, exposed to the red blood cell, and the red blood cell then gets modified as a result. So for any texts that were around in the 80s and 90s, you would probably know exactly what to do because this was something that was done far more commonly back then. And really the only answer in this, there's no other way around it, is that it's acid. And so what happens is that amine group that you have, that NH2 group that's sitting off to the side, with your acetyl group that's floating around in solution, if you add acid back and you get the pH down to less than 6.5, you can actually reacetylate and get this to add back on, which means that it looks like a group A again, and therefore your reactivity will go away. Your group B, acquired group B reactivity should go away. Okay, so now what I did was I instructed the text to use acid, and we can see that column 1 here is neat, column 2 is with hydrochloric acid, 3 is with PBS. Now, typically, when you're going to do this, you would use acetic anhydride, but you can use hydrochloric acid. You have to use a little bit of a higher molar concentration than what we use in the lab, and that's the reason why there's this just slight discrepancy here. But what we have is we've got the 3, no glutination, 2, 2 plus agglutination. We had acid, no glutination, no glutination, no glutination. As I said, 1 plus, it weakened, but it didn't completely go away. When we look at the PBS control, it's 2 plus, 2 plus, 2 plus. So the PBS control is valid, our hydrochloric acid worked, and it largely neutralized the acquired B, except for this, like I said, this one guy here. So then the question becomes, what else can you do to resolve it? And this is a less commonly used method, so I don't know if we have any texts that may have worked in the 60s and 70s, but back then, they also used to do something else. And again, it's just, you just have to know it. It's using galactosamine. And the reason why they did this was, if you're adding acid to your tube, you actually enhance, and I mean you can really enhance, autoantibodies, especially the colds. And so you can have an autoantibody that would normally react only at immediate spin, and now you have it reacting at 37 in IAT because of the addition of the acid. And so the use of galactosamine was actually done to avoid the enhancement of autoantibodies. And again, we'll talk about this in the next few slides, but most of the reagents that are out there now have acidified buffers in order to prevent all of this. So they add acid already to the monoclonal, so that even if it were to happen, for example, like in this case, which is uncommon, the antibody should theoretically not react that way because it's in the presence of acid. So now let's talk a little bit about these monoclonals that are causing the acquired B phenotype that we see by tube. The first one that was described was the ES4 antibody, and that's the monoclonal name, so it's nothing special. It was first noted in 1992, specifically when this ES4 came out, and it was used by Gamma, BCA, and Immucor, but not Ortho. And a number of reports kept coming out year after year, and so essentially the companies, as I said, added acid back into the buffer solution of these monoclonals so that they had acid plus the antibody, and therefore they would not get acquired B. The ES4 antibody's been phased out anyways, so it's not used anymore, which is why it's pretty uncommon to see an acquired B, and that's why I wanted to make sure I did this case. It's pretty darn uncommon now with the new monoclonals that are out, and a lot of the companies will include some screening for acquired B, and as I said, they also acidify their buffers, so this is not something that you should see very often. And let's get to the next slide where I talk a little bit more about the monoclonals. So as an antigen, the acquired B occurs because of decetylation, and this can occur from Clostridium or E. coli. Those are the two most common ones that are usually reported in the case reports where people noted that there was an acquired B phenotype. It only happens with group A individuals, right? Because this decetylation enzyme only works on the GalNAc. And remember, GalNAc is the added sugar for the group A antigen. Therefore, group O and group B cannot be affected by this enzyme. It's actually seen more often in the A1 versus A2, and it's because the A2 has less antigen present and the A antigen is slightly modified, and so therefore you don't see it. You need to have enough of the antigen on there for the deacetylation enzyme to act, and then for you to see it by monoclonal. And the reactivity strength of the anti-B that you see of your monoclonal is proportional to the A on the surface of the red cell. So with an A1 person, you've got a lot more A than an A2 person who has fewer A. So when this gets converted to a B, it's easier to see it by monoclonal, whereas if this gets converted to a B, it's a lot harder to see it by tube. And there was some speculation back in the day that the anti-B from an A2 individual actually could detect an acquired B better than an anti-B from an A1, but it was never really held up and it was never really fully fleshed out. But you may hear that every once in a while, that an anti-B, naturally occurring anti-B from an A2 individual, can detect acquired B, whereas an anti-B from an A1 individual should not react with an acquired B. So when we're talking about the antibody, as I said, you acidify the plasma, usually to around pH of 6, and it prevents the agglutination from the acquired B. Acquired B can potentiate cold agglutinins, and so it can falsely cause your sample to agglutinate, making you think that there's an alloantibody in there. And it can even make group O and group A agglutinate if you add enough acid. The addition of the 1.01 molar galactosamine with essentially a physiologic, maybe slightly higher than physiologic pH, can disperse the agglutination, and you don't have to worry about the cold agglutinin issue. Now, you can form an anti-B in your plasma, or you should have an anti-B in your plasma if you're group A, and it reacts with all normal group B red blood cells. It does not react with your own red blood cells, and it can be inhibited with group B saliva. So if you're group A, you then become an acquired B. You already have an anti-B in your plasma, right? And so this anti-B does not react with an acquired B. And it does not react with your own red blood cells, which is essentially acquired B. And that's what's unique about this, right? Your own antibodies don't recognize this acquired B phenotype, but they'll react with everything else. And your antibody can be inhibited with group B saliva. When we talk about the clinical significance, there have been people that are A1 phenotype that have been transfused with acquired B. Excuse me, there are people who were acquired B who received A1 red blood cells, and nothing happened. What they actually found was that those group A1 cells actually became acquired B in vivo. So they get converted because the enzyme's floating around in the plasma, and it just converts the A1 into acquired B. And of course, when you're transfused group O red blood cells, there's no issue because the group O can't be converted into an acquired B. This ends our Tier 2 answer session. Sorry if I rambled a little bit. The acquired B has a lot of basic science behind it, so if there are any questions at all, please bring them to the live session, and I'm more than happy to chat then. And the next one coming up, hopefully in a few days, will be the Tier 3, and I'm looking forward to seeing everyone at the live session.